Hello everybody, I am Akerke from Accessibility Club and today I am going to go through the questions on topic 6.5 Neurons and Synapses from Unit Human Physiology that can appear on your exam or assessments. This video is aimed on explanation of the topic after you have covered this topic on your biology lessons, and the explanation of the topic is by using examples from the questions from assessment papers. Let's start. So, the first question can appear in paper 1. It is a multiple choice question that says atropine drops are used by opticians to dilate the pupil so that a thorough examination of retina can be performed. Atropine binds to acetylcholine receptors in synapses. The question is, what is the effect of atropine binding in synapses? The task already explains the principle of atropine work in synapses, since, as we know, acetylcholine is a neurotransmitter that is responsible for transferring the impulse for muscle to contract. So, we can suggest that when acetylcholine receptors are blocked by atropines in synapses, it prevents the contraction of muscles that constrict the pupil and leads to pupil dilation. So, we know that acetylcholine receptors are located at the postsynaptic membrane in synapses, since acetylcholine is released from the presynaptic membrane. Knowing that, we can say that variant C here is most suitable for this question. And it is correct since it says that atropine prevents binding of acetylcholine at the postsynaptic membrane. So it blocks the acetylcholine receptors, which means that muscle contraction do not occur and the pupil is dilated. So let's move to the next question. The image shows a neuron. Here's a picture with a better quality. And the question is, what is the function of structure X? As we remember from the neuron structure, this part is called myelin sheaths. Thanks to them, the impulse propagation is much faster than without them, and it is easier to understand by looking at its animation. See? The myelin sheaths help impulse to move faster through the axon, so it just jumps over them. So, of all the variants, A is the most suitable, and it is also correct since myelin sheath increases the speed of transmission along the axon, and it is the main function of myelin sheath. Not all nerve cells have myelin sheaths, and so that we can compare the speed of propagation of nerve impulses with myelin sheaths and without them. Let's move to the next questions, and these questions may appear in paper 2, though it's extended questions that require you to write something and not just choose an answer from several variants. So the question asks, uh, the, the question weighs 2 marks and asks to outline the role of sodium-potassium pump in maintaining the resting potential. 2 marks means that you must mention two main things about the significance of sodium-potassium pumps to get a full mark. Exactly, you should say what do sodium-potassium pump do. They move sodium and potassium ions against their concentration gradient by active transport, so it requires energy. You can also mention the required ATP hydrolysis, but I don't think that it's the main point here. Then, this creates an electrochemical gradient or concentration gradient, as you want, whereby the cell interior is relatively negative compared to the environment outside the cell, so that the facilitated diffusion can occur during the further action potential. So we know that firstly we have resting potential, then action potential, which has depolarization, repolarization, and refractory period. It is not necessary to mention all the details since the common term outline requires a brief account and summary, and these 2 points are enough. So you must mention that sodium potassium pump create a concentration gradient because they move uh, sodium and potassium ions against their concentration gradient, and uh, which can help further action potential to occur. That will be enough. Next question asks us to outline how the neuron is stimulated to release a neurotransmitter. Again, you need to mention two key points and in this process. As I said before, neurotransmitters are released from the presynaptic membrane on axon terminal. When an action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. Calcium ions diffuse into the cell to promote the fusion of vesicles that contain neurotransmitters with the cell, within the cell membrane. Mentioning the opening of calcium channel and or diffusion of calcium ions into the cell will give you 1 mark. Then you need to write that neurotransmitters are released from the axon terminals by exocytosis, bulk transport. It is important to use such terms as vesicles and exocytosis because it is the principle of this process, and these terms are very important in describing this process, even in outlining this process. So you are more likely to get full marks for correct terminology used. And then you can write that neurotransmitters bind to specific receptors at the postsynaptic membrane, but it is not necessary to mention since the question asks about the release of neurotransmitters, but not the synaptic transfer itself. So you should just write what happens in presynaptic membrane and what is released and how it is released from the presynaptic membrane. And that would be enough, since the question weighs only 2 marks. Next question. It is very similar to the first one, to the very first one that is from paper 1. It asks to explain the action of neonicotinoid pesticides in insects. The common term explain means giving detailed account or picture of a situation, event, pattern, or process. Here, the question requires a detailed explanation of action of neonicotinoid in pesticides. Pesticides are chemical compounds that used to kill pests, including insects. So, neonicotinoid, as well as a previously mentioned atropine, bind to the acetylcholine receptors in synapses, which means it blocks the acetylcholine from binding to the postsynaptic receptors. To get a full mark, you must say about the blocking of acetylcholine that neonicotinoid binds to the active sites of acetylcholine receptors, which prevent the action of acetylcholine that is responsible for controlled impulse of muscle contraction. Why the impulse is controlled? Because the enzyme called acetylcholine esterase, which breaks down acetylcholine after its binding to postsynaptic membrane and helps the acetylcholine go back to the presynaptic membrane to avoid overstimulation and uncontrolled muscle contractions. When neonicotinoid blocks acetylcholine receptors, acetylcholine esterase is not able to break down neonicotinoids since it is not suitable to them because, as we know, enzyme is very specific to its substrate. So acetylcholine esterase can only break down acetylcholine and not other neurotransmitters or compounds that block neurotransmitter receptors. So the uncontrolled impulses for muscle contractions cause paralysis. When neonicotinoid blocks acetylcholine receptors, it can lead to uncontrolled impulses for muscle contractions, which causes paralysis. So insects do not die under the effect of pesticide with neonicotinoid; they just paralyzed, and it is easy to eliminate them. To be sure about getting full mark for this question, you should mention all these processes so that you will be able to satisfy the common term explain that requires details in your answer. Also, do not forget about terminology because it is really important in paper 2. If you use the correct terminology, you will get a higher mark for sure because it shows that you know what terms you can use in describing or explaining some processes. You can also face questions related to these topics that require your understanding of graphs. To answer them correctly, you need to memorize how the membrane potential, so membrane charge is as you want, uh, changes during resting potential, threshold potential, and action potential. This animation helps to understand this better. During resting potential, sodium and potassium ions are moved against their concentration gradient by active transport. Then the depolarization of active potential happens, and the potential on which depolarization occurs is called threshold potential. In this case, the question asks what is the approximate value of the threshold potential according to this graph. So in this case, it is minus 50 millivolts, and it is the answer to this question. But if we look at the graph, what we can see, what happens after threshold potential. When the potential reaches its highest value, the repolarization happens and the potential rapidly decreases then. When it reaches its lowest point, then the refractory period is happening, and after that, it returns to the level of resting potential. So that's all for today. I might have not covered all the types of questions that may appear on your exam or the material related to this topic, but anyways, if you'll have any questions, you can always email me and ask any questions you want regarding biology SL or HL program. Thank you for watching and good luck!